good afternoon to everyone. It's a really pleasure to be here to share a little of part of my experience during the junior visiting professor abroad. My name is Melissa Xavier. I'm professor of mycology, as Andrea told before. So I did my presentation in 4 steps that we are going to start now, of course, with some background. So similar to what Professor Pedro was talking before, our focus was in medical mycology. And the problem here is fungal disease. And how big is this problem? The global impact of invasive and endemic fungal disease is huge. And this occurs mainly because of this disease are underdiagnosed in a global aspect. Then we need everyone to work to reduce the subdiagnosis and to improve the treatment, the correct treatment of this disease. So these 2 organizations are the main organizations in this field nowadays. In the last 2 decades, the fungal disease acquired this important impact in human health, and it's still underdiagnosed. And this is our goal. When we talk about fungal, when we talk about infectious disease, it's common to people think in bacteria and virus and forget about the fungi. And when we talk about mycoses, it's simple to think in superficial and cutaneous lesions, and it's not what we were working. We are working in a huge and severe disease that has a great impact in human health, that are the invasive and opportunist and endemic fungal disease, severe mycoses. Then we know that nowadays it's estimated that over 500,000 HIV patients die of fungal infection every year. Over 10 million of people live with fungal asthma. And according to what Pedro told us before, it's interesting that it's microbiology, we work together, and it's completely know that the increase of the diagnosis of fungal disease will reduce the antibiotic use, the misuse of antibiotic, and then we are also contributing to the multi-resistance of the against these antibiotics. So this is an important field in human health that we need to work, and is our focus today. So this is an illustrative panorama of what we are talking about. There are a lot of group of risks that can be affected by these invasive and opportunistic fungal infections that can progress to death. And then this global actual fund for fungal infections, this organization suggests that if we can work improving the diagnosis and the treatment of 60% of the patients that need, we could save over 1 million of lives in the next 5 years. So we need this field urge for more research regarding this. And bringing this information from global aspect to our national aspect, we can see here an estimative made only with the notification of the disease, and we know that these fungal diseases are not officially reportable disease. So for sure, this is subestimated. But we demonstrate that more than 3.8 million of people in Brazil may suffering from serious fungal infections. So this is our problem. And here we are. I have the pleasure and to coordinate the mycology lab of our medicine faculty of our institution. And we work exactly in this goal. I have a great team, and this is the way that we can do the right and the better things with people that are really really working hard in the same goal and working together. We have an interdisciplinary and multi-professional group team working in this same objective. And the main objective, so, is our main goal is think fungus, save lives. So when the opportunity to participate in the PrInt appeared, Professor Andrea was my partner in this, and we could see that I could work in the human health in coastal ecosystem with this field that I told for you, in the sub-project prevention and control of disease and illness that affect human health and quality of life in coastal ecosystems. So the researcher that I made contact was Professor David Stevens, and I applied for the junior visiting professor grant, and I have this great opportunity to be selected and participate in this program, different of both of the other professors that talked before. I had the privilege to was there in previous the pandemic period, so I really could work all of the periods in the USA. So from Rio Grande, I went to California, to California Institute for Medical Research. It's located in San Jose, near San Francisco. And I choose to work with David Stevens, and I have the honor to receive his accept to receive me there to work with him and his team. He's an important researcher in the medical mycology field. He has a huge experience in this fungal disease. He is the president of the California Institute for Medical Research since 2001, and he is emeritus professor of infectious disease in the Stanford University. He received a lot of grants and honors as American Academy of Microbiology Fellow, Rhoda Benham Medal in Medical Mycology Society of the Americans, another medal for the International Society for Human and Animal Mycology, and award of a coccidioid mycosis study group. And he was also the project leader in the NIH mycosis study group for 10 years. So here we can see that he could and is still contributing a lot with us in this field of the medical mycology. He is also the co-chairman of one of the mainly conference about aspergillus and aspergillosis, And I had the pleasure to be in this Congress in 2008 when I was a PhD student. And it was interesting because when we were changing emails to schedule and to organize the visiting professor period, he sent me an email that I thought your name sounded familiar. Didn't you get a scholarship? So he was one of the co-chairmen that signed my scholarship in that occasion. And I have no idea why he recognized my name, but he recognized it. So I went there with the project Diagnosis, Prevention, and Control of Invasive and Endemic Mycoses in the One Health context, that aimed to establish partnerships with international researchers of excellence in the medical mycology field, contribute to the internationalization of our institution, share experience and transfer technology, which would contribute to the human health promotion, emphasized on the diagnosis, prevention, control, and treatment of invasive and endemic mycoses, contributing to human health in coastal ecosystems in accordance with our sub-project of the PrInt in human health in coastal ecosystems. So I stayed there for 6 months, and the end of the period, it was exactly on the start of the pandemic. So I had lucky to be there in the last period that I could work all the time in a presential mode. It was from September 2019 to March 2020. We had weekly meetings with the lab. And of course, starting the beginning, I had the opportunity to show and share some good things of our culture. I even take there the chimarrão, and everyone toast the chimarrão there, and to share good things of our institution with the medical mycology team of Professor David Stevens. That it was the great one of the great experience that I could share on day by day cosmopolitan environment. So a lot of people with different cultures and all working together. So in that period, we developed some experimental studies and mainly focused in antifungal drug discovery. We made an in vitro and in vivo studies, mostly regarding 3 new potentials antifungal drugs. These fields are really important because, as you know, we have scarce, limited number of antifungals available in the market to treat fungal disease, and almost all of them act at the plasmatic membrane of fungi, and we have few of others. The green ones are the ones that are available to use, few others that act in other sites but are not really available, and it's less used. And the newest antifungals class, that is echinocandins, that work, acts inhibiting the cell wall of the fungi, but it's too expensive. But it's the newest and that has low toxicity because fungal are eukaryotic cells, so it's difficult to find antifungal drugs that are not toxic to the hosts. Then we worked with nicomycin Z, that it's a chitin synthase inhibitor, then acts in cell wall also. It's a good candidate to a new antifungal drug. It's already in some clinical trials studies against coccidioidomycosis, but only against this mycosis, not the others. So we worked in this field. And the other two potentials antifungal drugs that we studied, it was gallium and diphenyl diselenide. These works were developed against the fungus genus Aspergillus, Sporotrix, and Coccidioides. To work with Coccidioides, we used always the BSL-3 because the high virulence of this pathogen, and this was available at the institute. Here are some pictures. We have really hard but good work there. And besides the experiments and the research that I could develop there, Professor Stevens was also very compromised with my period there to increase our visibility. And then he scheduled a lot of networking meetings with important researchers in the field of medical mycology. That was a great opportunity to improve our work. So one of these that is our partner now, it's David Larud. He is the CEO of the Valley Fever Solutions, that are the company that produce the Nicomycin Z. And so he were working with us in all the period that I was there, providing the drug for our in vitro and in vivo experiments. And he also send us and give a donation to us to continue doing work here with these drugs that is really promising. And he is already a partner. Another important network meeting that Professor Stevens scheduled, it was really, really interesting. It was with the Professor Ellen Jo Baron. She's a really kind person, and she is from the Stanford University School of Medicine. She worked at the NIH clinical laboratories, at the CLSI, the CFAID, and she also has a really interesting program that she was the co-founder of this program, that is the Diagnostic Microbiology Development Program. Is focusing in educating research poor nations on laboratory microbiology. So we can see that in Stanford, of course, they have all of the good, great, and the highest and newest equipments to do their works, a lot of technology. But this program that she helped to develop and she were doing, it was to go to research poor nations and to teach the classical microbiology, because with classical microbiology, and this of course includes classical mycology, we can start saving a lot of lives and doing a lot of diagnosis. So we don't need to have all of the top equipments to do this. So it's in this focus, the program. I had the opportunity to discuss with her a lot about this program, and it's really, really nice. It's a beautiful work. And then we have some dinners together to talk about, and she guided me in a technical visit to the Stanford University Medical Center, to the clinical labs of the Stanford University. And as she was the chief of these labs for around 10 years, she was a very welcome person there. And as I was with her, I was a very welcome person there in this occasion. So it was really nice to see. It's a beautiful building. We cannot say that it's a lab because it's huge and a lot of labs, and with all of the equipments that we can imagine. And she also gave me the opportunity to work a week with the mycology team of the Stanford lab. So I have a great opportunity to learn, to discuss, and to share experience regarding the diagnostic routine in medical mycology in an institution of excellence abroad. And it's really interesting to know, to see, and to feel that, of course, sorry about that.
Your timer?
Sorry about that. No, the telephone is, the computer recognizes the telephone that is ringing here, sorry. And then it's really interesting to see that without all of this technology, we are able to do a lot of things here. So we do our best, and we can do a very good job without all of this technology. So it was a great time there with them to improve some procedures that we do here in our routine. Another important researcher that Professor Stevens scheduled meetings, it was Professor David Denning. He is, as this biography say, he is a prominent international face in the field of medical mycology. Everyone that works with medical mycology in the world knows him because he goes to everywhere to teach and to work in this main goal, which is reduce the underdiagnosis of fungal disease. And then he's the founding president of the Global Action Fund for Fungal Infections and the leader of the leading international fungal education that are these two companies. He's also the founder of the Aspergillus and Aspergillosis. He is the chief of the National Center for the Aspergillosis Treatment in Manchester. He's professor of infectious disease in Manchester. And then I met for the first time Professor David Denning in 2008 in this Congress that he is also a co-chairman. Professor David Denning was a fellow of Professor David Stevens before that. And then here in 2019, I had the opportunity to go to an informal dinner, and we discussed about medical mycology in this goal that is the goal of GAFI, that it's to ensure that by 2025, 95% of people with serious fungal disease are diagnosed and 90% are treated. So it's a big network that are engaged around the world in this goal. And another important partner that we could establish in this period, it was with the two main researchers in medical mycology field from the National Institute of Health from Portugal, Raquel Sabino and Cristina Verissimo. In this same period that I was as a visiting professor in the United States, one of my PhD students, Arizi, was was in a period of his sandwich with Raquel and Cristina, and this was, who opens this door was Professor Stevens. So we didn't know her before, and he made this this presentation for us. And then Arisi was there in Portugal, I was in USA, and all of our great team were here in Brazil, and we were working together, of course. Then here with these partners, we worked mainly in the One Health concept, specifically regarding molecular epidemiology of Aspergillus fumigati and azole resistant of Aspergillus fumigatus stricto sensu. So we are easy worked in his thesis with clinical isolates from humans, with clinical isolates from penguins and other marine birds, and with environment isolates in this context. And it was really nice, and now we have another projects with this group. And to finish these meetings, just to show to you that another interesting meeting that I could participate, it was a conference about coccidioidomycosis, a lecture that Professor Stevens were talking. But it was really interesting because it was in the Bay Area History of Medicine Society that was formally instituted in 1941, making it one of the oldest independent regional groups devoted to the history of medicine and health in the United States. So I had the opportunity to be in a dinner with a lot of researchers and from different fields of health. It was really interesting. Thus, we have, of course, achievements that I want to show and share with you. In the field of gallium metal that we worked as a new antifungal drug, we presented a poster in the 9th Advances Against Aspergillosis that occurred in Switzerland. And we received, our poster was an outstanding poster with this abstract, so it was really interesting. Regarding nicomycin Z, we had recently received the publication of this paper that we worked with, and this was an in vivo experiment that we developed there in the CIMR, and we published in the Antimicrobial Agents and Chemotherapy, and also we had 2 more papers that were published in good scientific periodics in the medical mycology area regarding nicomycin Z. One of these, before talk about this one, it was nice because for me, it's the same time that I have a paper published in a scientific periodic that is classified as A1. I never had this before, so this is what we were talking about: improve the quality of the research. And this other one paper, the first author is Vanice Poester. She was my PhD, one of my PhD fellow. And now she is a doctor and a colleague. And this paper was awarded by the Ira Salkin Memorial Award that promote award for the better paper published during the year in the medical mycology periodic, that it's an important periodic in the field. So Vanici received this award for this study that we developed together. And regarding innovation, we also had a patent deposition in the last month. So we can be sure that we are improving our quality, and this is the achievement of one of the achievements of PrInt. Regarding diphenyl diselenide, we also already had 4 papers published. All of them, you are going to see that David Stevens is with us. He has been a great partner since then. In the One Health concept that I told to you, that we developed some experiments with the partners, with the partnership of the National Institute of Health from Portugal. We also had 3 papers published in this context, and Arisi as the first author, that was my PhD fellow in that period, that were there. And this was the main of these papers that resume everything, we can say that. And it was really interesting. It was published in a good scientific periodic, and it focused exactly in the azole resistance of Aspergillus fumigatus and the One Health concept. So we could show here in this paper, it's a review, we could show the relation between the fungicidal used in the environment, in the agriculture, the aspergillosis in the avian aspergillosis in birds and in marine birds, and the potential of these birds to carry these microorganisms, and humans involved in all of this circle. So it was a really nice paper, and I had already a return about that. Last month, I was invited to provide a lecture in the post-graduation program of infectious disease of the Federal University of Espírito Santo to talk about this focus. It was Aspergillus and aspergillosis in the One Health context and the azole resistance. So it's really nice to see that we are improving our quality and visibility. In the One Health context, but mainly in the diagnostic context, we also had improvements already. This was a recent paper published from our group, Vanici, that is the first author, that it was the PhD, it's our colleague that work in the respiratory causes field. And it's really nice because it's a work that we have been doing since 2010, and we have been improving this work as along the years, and we could now establish an assistance service for human respiratory causes in our city. So here we have a service for our community to improve the diagnosis and the treatment of human respiratory causes. And we had beautiful results when we compared the previous period of our service with post period of our service. And we were very glad with these results, the social impact of these results. And Dr. Stevens was essential to help us to put all of this importance that we were glad just in the social way to put it as a research paper that was published in a very good periodic, scientific periodic. So we still improving also the quality of our diagnostic. So we are achieving better service to the community regarding the human health. So here another papers that were case reports of fungal infections, invasive and endemic fungal infections diagnosed in our lab here after the visiting period abroad, and with the participation of David Stevens in all of them. And then I can say that the internationalization program for me allowed to establish partnerships, improving quality of academic production, institutional visibility, possibilities for students' mobility, experience shares, technology transference, engage our lab, representing our institution, into international research networks in the medical mycology field. So concluding, for sure, I wanna thank for this opportunity that was really, really good, really, really impressive. I'm sure to say that I was one person before, and after this life experience and professional and personal life experience, I'm different now. It was a great opportunity. So I want to thank everyone who worked hard to bring this program to PrInt-FURG and made the possibility to us to participate on this. CAPES, for sure, our post-graduation program, all of our partners, the Valley Fever Solution, and for sure, Professor Stevens, that I was very welcome there, and it was a great period. He could propose a lot of things important to me and all of the team that worked with me there. I for sure need to also thank my group, my research group, that I couldn't do everything without all of the great heads that I have with me. We are really a multi-professional and interdisciplinary and good researchers. I'm surrounded of great researchers, and I'm glad of it. Thank you, girls and boys. Now we are almost all of us are girls, but thank you for everyone. Thank you, Andrea. You are one of the really important person, mainly in the beginning, to help me to apply to this program and to have this opportunity. And for sure, I want to thank also to my family that were my strength and my main partners before, during, and after all of this period. Concluding, the perspectives is bringing back and share all of the luggage acquired during this period. We need to continuously improving always. Science is a lot of things together, and science is much better when we have more partners. As more good partners we have, as more science will be. So with all of this, I want to work creating a healthier future, and to leave a take-home message I want to share with you. I had the opportunity to visit the Big Basin Redwoods State Park in California during the period that was there with all of the team, part of the team of the medical mycology lab of Professor Stevens. It was really beautiful to see that we are such little things are surrounded of these giant trees. And I think everyone had known that in 2020, almost all of these forests burned. And then the take-home message is there in 2021 now, even the trees that were totally burned are re-emerging. So in this morning, Professor Eduardo showed a table for us. There, I didn't ask, sorry, Professor Eduardo, but I stole from you to put here, because I think we are going to need to finding a way as the nature find their way to re-emerge, because we didn't burn it with fire, but the pandemic was also devastating in terms of imperative to promote more opportunities like this one that I have. Thank you for your attention. And I finish here.
Thanks so much. It was almost 44 minutes, okay? It was in English.
Fine. That's all right, Andrea.
Sorry, Andrea.
It's in English, it was in Portuguese, it was 2 hours, so I'm glad that it's in English presentation.
No problem. Melissa is closing the afternoon session with a golden key. It's very touching and impressive how you can use a very small grant, really, and change and make a huge difference for your research, for your graduate program, for the medicine faculty, the School of Medicine, and it's impressive that you made so much in a very short time period, and all those connections, and it makes very clear that this program should continue. It's a very good example that I hope CAPES have the opportunity to see this presentation. I hope that it's going to make a huge difference for the graduate program in health science, because if this knowledge acquired abroad is shared with your colleagues, if this experience and this example is also seeked by other colleagues, professors, and students, because here they saw a very good example of how important it is to make connections worldwide and how it can improve our science. And it's a very applicable science, this applied science to look after the people that pay our salaries and maintain our university. It's a very good example of the return to the society. Thank you very much, Melissa, and thank you for all the speakers. I'm very glad to see all these achievements. Thank you. Andrea, please carry on.
Okay, thank you. Well, Melissa, she knows I told her that, and I fill up the report, the mid-term report, and I told her that she is a case of success, a success case that you can that PrInt represents, because she didn't have the opportunity, she didn't to have a long time abroad, and this is the first opportunity, and she profits and make the difference, and this is the most important. So congratulations, and I'm sure that you spent this time because you had a lot of things, nice things to show us, and it was a pleasure to see you after I make a joke. It's common. Okay, so, but it was very nice, your presentation. If someone wants to make a question or a comments, you know that it was a long day, but someone...
Can I talk?
Yes, please.
Yeah. No, I just like to congratulate Melissa. It was extensive but very interesting presentation, and you made the most. And it seems to me that you made the most of this opportunity, and you are bringing, you and Pedro bringing the improving the science make at PrInt-FURG. And the topic is very interesting, very far away from my area of my specific area, but there are very much interesting things that it's interesting for everybody. That's such a issue, that health issue, that environmental issue that's interesting for everybody. And can I ask you just a technical question about this thing azole? While you were talking and it showed the title of your paper, I just look it quickly at Google, because azole for me, it's another thing, it's a plant.
All right.
I am a plant specialist, so I went to look for what was that. And I just saw there's something that we humans metabolize it also, that's used against the fungi. Could just quickly just explain that, what the thing is, just to clear, just make it like a useful question.
Perfect. It's a great question, because azole is the main group of antifungal available to treat fungal disease, to treat since onychomycosis to invasive candidemia, that it's candida in septicemia by candida. So we use the same group of antifungal, that it's azoles, miconazole, fluconazole, itraconazole. Some of them for sure you have listened about, and women that have a lot of vulvaginity by candida, the same fluconazole.
Now I remember, cetaconazole.
Yeah. Cetoconazole, yes, all of them are azoles. And the main problem in the discovery of new antifungals is that fungal cells are eukaryotics as us, as our cells. So it's difficult to find a drug that it's toxic to the fungal cell and it's not toxic to the host. And then what is the problem now that it's emerging globally is that some microorganisms are developing resistance and cross resistance between the drugs. So, and pathogen that develop a resistance by itraconazole, that it's available and it's widely used, also develop resistance for voriconazole, that it's a topic antifungal that is used only to severe cases of aspergillosis. Then it's a way that we need to work to find antifungals that act in different sites of action, in different sites, not in the plasmatic membrane, not in the ergosterol, that it's the main component of the plasmatic membrane of fungi. And this is the idea, to search for new antifungals that act in different sites of the fungal cell. We have, maybe you have heard about Candida auris. It's another new species of Candida that were discovered in first time 2009, so it's recent, and it's a multi-resistant fungi that when cause septicemia, fungemi, it's really difficult to treat and the mortality is very high. We have a lot of aspergillus now that are developing resistance, and a lot of the the fungicides used in the agriculture are also based in are also from the azole groups, epoconazole, and then the fungi that are in the environment is the same fungi that can cause disease in humans and in animals, and we can develop the resistance in the environment and then infect some hosts already resistant to the antifungal that is going to be used to treat. I don't know if I could answer you.
Oh, that's very interesting, very much interesting, because you answered lots of questions here that I had, for example, in mind, that why this kind of fungus is more difficult to treat than bacteria and virus. I am a biologist, so I understand the tree of life and what does it mean, eukaryotic and the prokaryotic. So I always teach that, but I never thought that the problem with fungus and us is because we are both eukaryotic. So it's very interesting. That's for me as a biologist, I never realized or I never knew that. And this thing about the candida that said that's what's called the super candida.
Yeah,
that was
super fungus.
Yeah, appears in the northeast, in some hospital in northeast Brazil this year, actually, or beginning of this year. Some people died of it.
As an outbreak.
Yeah. Okay, very interesting. Thank you very much. I will remember that.
Hey, Margaret. She's looking for the candida in the water that you are collecting together, the PrInt project. She is also working with us.
Yeah, the one that you explained to us, is that, Andrea?
But you are working with bacteria, and afterwards, when she knows that you are doing this, collect the water in different sites.
So have you found?
Today it's the third.
Not yet.
Not yet.
Not yet.
That's good, at least.
Yes. Yes.
Oh, that's very interesting. Thank you very much. You clarify very much.
Thank you. Well, so thank you. I think that
I see that our international Secretary for International Affairs is still with us. Thank you, Miltinho. We'd like to
You're very welcome.
to provide some words because it was very nice to see all these presentations.
Just one word, Eduardo. I do believe that to reach real internationalization, you need both things: the international cooperation and internationalization at home. No, today you have a nice demonstration of both. No, all the experience exchange, academic exchange, cultural exchange, cooperation abroad with different institutions. Very nice information, which makes the international cooperation a reality. And this workshop is an example of the internationalization at home. No, it's for me, from the Reinter perspective, watching this all day long workshop with such a nice information, everybody in English, you know, and it's a great advance in terms of our internationalization. So my congratulations to all of the speakers. It was very, very nice. Thank you.
Thank you, Professor Milton. Yeah, and thank you again for everyone for all the speakers, and thank you for sticking together until almost 6 p.m. Tomorrow we restart at 9:30, right, Andrea?
Yes, and I will send a new link for tomorrow by email to everyone, because this link is only for this meeting. So we will receive tonight, a new link for tomorrow.
And thank you especially to you, Andrea, for all the dedication to this program. And today, Andrea is officially director of graduate program. So welcome to this new position at ProPESP. Well, see you tomorrow, everyone. Have a good evening, and thank you again. Bye.
Thank you for the nice.
Thank you. See you tomorrow. Bye-bye.
See you tomorrow. Bye-bye.
Thank you, everybody. See you
tomorrow. Bye-bye.